So before we start with this session, just a brief introduction on the Young Investigators United, which may be well known for most of you, but if not. The EASO's Young Investigators United was actually established in 2005. The young investigators then involved were Tommy Visser and Kirsi Pietalinen. And since then, the Young Investigators United have actually a scientific session combined with a social event annually at the ECO congresses. And the main aim of the Young Investigators United is actually to get to know the people working in the field of obesity, young investigators, to exchange ideas, to share expertise, and also to get to know each other and maybe to initiate some collaborations. So today we will have several presentations during this scientific part of the session. 2 nominees for the Best Young Investigators United Thesis Award. So that are Claudia Sikorski and Doreen Segers. So they were both theses were selected based on several theses that we received. So both of them will briefly explain to you and present their data. And at the end of this session, one of the 2 will be announced as the winner. And we also have, and we're very happy that we have some lectures about funding possibilities within Europe. So we also have representatives from the European Research Council. So we're very pleased, and we welcome them that they're here. And also Maria Bryant, who also received some funding, and she will also present some things related to funding possibilities. So let me start by introducing EASO's president, Professor Gemma Frubeck. She will just spend a few words on the opening of this session. And Gemma is actually president of EASO, as I said, and she combines the everyday work in the clinic with her task as head of the department of the Metabolic Research Laboratory at the University of Navarra in Spain. And in 2009, she became president-elect of EASO, and since May 2012, Gemma has actually stepped in as president of EASO. So I don't spend too much words, and I would welcome Gemma to open this scientific session.
Okay, thank you very much. While the slides are coming up, I would really like to thank the organizers, in this case the Young Investigators United, to letting me share a couple of thoughts with you. And you know that when we did our strategic review or strategic analysis, for the first time, we really asked the participation of the Young Investigators United via your board, and it was the very first time, and it was really one of my very important thoughts that you also have a saying, because you are not really the future of the society, you are the present of the society. In fact, you should be already looking for the future in the younger generations, because we want to involve you in all of our decisions, because it will make the society much more dynamic, it will get new ideas, and it will evolve to what we really want the society to be, so that we are really updated, innovative, and that we have fresh ideas and young blood. In this sense, we had our executive committee meeting at the beginning of the Congress on Saturday, and one of the Young Investigators United board members was represented there, and we thought it's important to have you involved not only in the sessions like this one, but also in how the society is run, because sometimes you tell us what your desires are, but it's not that easy to put it into practice, because, well, we would also like to have more travel grants, more money for young investigators, but the situation is not that easy, so that you know everything from inside and that you know how the society is run. And I will put this slide, which I think illustrates very well what the passing of the torch should be. This is a statue that is in front of the medical school of the University of Madrid, the Universidad Complutense, and it depicts the old guy, which is down here, which is the old guy just living without forces and without energy, and passing the torch to the younger guy, which is already on the horse and ready to depart. So we want to do this also at EASO, and you are the guys that are on the horse and the ones that will have to follow up. And for this reason, we are very excited that in the opening ceremony we had excellent Young Investigators Award presentations. So that warrants the future of the basic, the clinical, and the public health research. We also have this session, which is very important, with the thesis presentation. And our next step will be also to include a young investigator in each of the task forces, so that you are also having a word there and getting to know how the different task forces are run and have an impact also on that side. So I wish you a very fruitful and productive session, and that you really appreciate how important it is to have this kind of session. I would have loved to have such session when I was a little bit younger, and to have all the advice of the people coming from the ERC, to have the opportunity to share with our colleagues, to already initiate networking. You really are going to be very fortunate to try to take this into the next years and also into a very rich experience for you. So thank you very much.
So thank you, Professor Frubeck. During this session, we will have the pleasure to have the presentation of the two candidates for the Young Investigators United Best Thesis Award. And the first candidate for the Young Investigator Best Thesis Award is Dr. Doreen Segers from Belgium. In 2007, Doreen Segers obtained her master's degree in biomedical sciences at the University of Antwerp. She then started her doctoral fellowship in the obesity research group at the Department of Medical Genetics at the University of Antwerp, supervised by Professor Wim van Hul and Professor Luc van Gaal. And this fellowship was funded by a PhD grant of the Institute for the Promotion of Innovation through Science and Technology in Flanders. The purpose of this PhD project was to unravel the involvement of several genes from the leptin-melanocortin pathways in the regulation of food intake and energy balance. In 2012, she obtained her doctoral degree in biomedical sciences, and she recently started a postdoc fellowship in which she will also expand her research towards the molecular genetic aspects of the pathogenesis of non-alcoholic fatty liver disease. Now she will show us her presentation entitled Molecular Genetic Analysis of Genes from the Leptin-melanocortin Pathways in the Pathogenesis of Obesity. So please, Dr. Segers. I think it doesn't work.
Okay, so thank you for that kind introduction. First, I would like to start by thanking the Scientific Committee and, of course, the EASO Young Investigators United Board for selecting me as a finalist and for giving me the opportunity to present my work here today. I'll start with an overview of the presentation. First, I'll give an introduction about the genetic factors involved in obesity, and then a short introduction about the leptin-melanocortin pathway, which was the subject of my research. Then I will show you the results on 4 candidate genes from this pathway, and I will end with some conclusions. So as you all know, obesity is heritable, which means that part of the phenotypical variability is caused by genetic factors. Twin and adoption studies have been carried out in order to estimate this heritability, and it was estimated that BMI and weight are up to 70% heritable. This means that in the general population, an interplay of genetic combined with environmental factors will determine an individual's BMI. When we study the genetics of obesity, there is an entire spectrum, and on the one end of the spectrum, there is common obesity that occurs in the general population and that has a multifactorial cause. This is also called complex obesity. On the other end of the spectrum here, there are some rare cases of severe familial inherited obesity, which is caused by single gene mutations and is called monogenic obesity. Monogenic obesity is studied by performing mutation analysis on selected candidate genes, while complex obesity is studied by performing association studies, either candidate-based or genome-wide, and both strategies were used in this project. So now the leptin-melanocortin pathway. This pathway is the most important hypothalamic circuit which is involved in the regulation of energy homeostasis and food intake. And this pathway functions as an endocrine feedback loop. This is a schematic overview. And as you can see, the adipose hormone leptin will circulate in relation to the amount of adipose tissue in the body. In the brain, leptin will bind its receptors, thereby causing an increase in the expression of POMC and CART proteins. POMC will then be cleaved into biologically active melanocortin peptides, such as alpha-MSH. And these melanocortins can then bind the melanocortin 4 and also the melanocortin 3 receptors, which will then become active and regulate energy homeostasis by decreasing food intake and decreasing feed efficiency. Mutations in a number of genes from this pathway have been shown to cause monogenic obesity, and these include mutations in leptin and its receptor, but also mutations in POMC and, of course, the melanocortin 4 receptor have been demonstrated. So as we know that genes from the key players from this pathway are involved in causing monogenic obesity, less is known about the downstream genes. And the aim of this PhD project was therefore to unravel the role of genetic variation in genes encoding downstream proteins from this pathway. So the first gene we studied was the MC3R gene, and for this gene we performed a mutation analysis and an association study. MC3R is located here in the pathway, together with the well-known MC4R receptor. MC3R is a 7-transmembrane G-coupled protein receptor, which is activated by melanocortin peptides such as alpha-MSH. Animal models have shown that MC3R defects can lead to an increased feed efficiency, increased fat mass, and increased leptin levels, and also hypophagia has been reported. We therefore hypothesized that defects in MC3R could also lead to early onset obesity in humans. We then screened the entire coding region of the gene in our cohort of approximately 600 obese children and adolescents and 400 lean controls. And in this mutation screen, we found a total of 10 novel coding variations. 6 of them have been identified in obese children, as indicated by the red squares, and 4 of them have been found in lean controls, as indicated in blue. For a number of these mutations, we performed functional experiments to investigate whether or not they affected the function of the protein. So as MC3R is a transmembrane receptor, we first performed confocal imaging in order to verify whether the mutated proteins were still present on the cell membrane or whether they were trapped intracellularly. And we also performed the Luciferase assay in order to investigate whether or not they could still activate the cyclic AMP pathway in response to stimulation with alpha-MSH. So here is part of the results. In the top row, you can see the results for a wild-type MC3R. MC3R is labeled with green fluorescent protein, as can be seen here. The cell membrane is labeled with a red stain. And here you can see the merged view where co-localization of the wild-type receptor can be visualized by the orange line around the cells. Now, as we look at the first mutation we characterized, again, we see an orange line around these cells indicating that there is co-localization for this mutant. Again, the same can be seen for the second mutation, again, orange meaning co-localization. However, for the third mutation we characterized, we found that it was intracellularly trapped, indicated by the green color inside the cell and the red staining of the membrane around it. So we then went on with Luciferase assay. Again, we found for the first two mutations that there was little or no difference in their ability to activate the cyclic AMP pathway. However, for the third mutation, we found a more than 40% decrease in its ability to activate the pathway. So these results, combined with confocal imaging, indicate that for this mutation, the leucine 299 valine mutation, this could be a likely cause of the obesity phenotype seen in this child. So to summarize this mutation analysis, we can say that we found multiple rare variants in the gene that are scattered throughout the gene and that do not seem to be exclusively pathogenic, as they appear both in obese cases and lean controls. It is therefore important to check conservation of the amino acids involved, to check familial segregation, if possible, and to perform functional experiments. Now, we also performed an association study for this gene, and we analyzed the 2 kb regions surrounding this gene. 2 tag SNPs were selected, one tag SNP upstream of the gene, not on this picture, and another tag SNP, which is a threonine 6 lysine coding variant, which is a proxy for this tag SNP, the valine 81 isoleucine variant. When we performed linear regression analysis, we found association of this coding SNP with weight as a quantitative trait in the obese population. Furthermore, a trend for association with obesity was observed. So to summarize, although we did not find a clear association with obesity, we did find the coding SNP to be possibly involved in weight regulation in obese individuals, which was confirmed by in vitro experiments where a functional effect for the double variant was determined. So we now know that genes from the upper part of the pathway are definitely involved in the pathogenesis of obesity, but less is known about the downstream part. So I'll briefly talk you through the rest of the project where these 3 genes were analyzed. We will talk about the BDNF gene, which is located there, the nesfatin or nucleobinin 2 gene located there, and the single-minded 1 gene located there. First, we analyzed the BDNF, or the brain-derived neurotrophic factor gene. For this gene, we also performed a mutation analysis on our population of obese children and lean controls. And we only found one coding variant in our obese population, the valine 46 methionine variant. And furthermore, also the common polymorphism valine 66 methionine was observed. However, as in silico predictions did not suggest a disease-causing role for this mutation, we can conclude that BDNF mutations do not play an essential role in the pathogenesis of childhood obesity. We then went on with a study involving the NUCB2 gene, which is a gene that was only recently identified. And we studied nesfatin 1, which is processed from the precursor protein nucleobinin 2 and encoded by the NUCB2 gene, and which was considered to be a novel satiety molecule associated with the melanocortin pathway. For this secreted protein, a role in energy homeostasis and metabolic control has also been suggested. So we started with an association study for this gene, and analysis of the HapMap indicated that genotyping of only 8 tag SNPs was sufficient to capture all 31 informative SNPs in the 75 kb region around the gene we selected. We then performed statistical analysis and found several SNPs to be associated with BMI in the male population. So when we look at the error bars, we see a recurrent pattern for each SNP, where the BMI is lowest in the homozygous group, is intermediate in the heterozygous group and is highest in the wild-type group, indicating a protective effect for polymorphisms in the NUCB2 gene in the male population. We also performed a mutation screen for this gene, where we screened the entire coding region, as indicated by the black bars in this gene overview, in our population of obese children and lean controls. In this mutation screen, we found 2 novel coding variants, being one missense mutation here and one nonsense mutation here. While no family data was available for the missense mutation, we found that the nonsense mutation was inherited from the obese father, making it a likely cause for the obesity phenotype seen in the proband and her father. So to summarize study on this gene, we can say that from the association study, we found that SNPs in NUCB2 might be involved in the protection against obesity in male subjects, which is in line with the hypothesis that NUCB2 might be involved in energy homeostasis. Furthermore, as far as we know, this was the first genetic approach to analyze the role of NUCB2 in the pathogenesis of obesity. Our mutation analysis led to the identification of the first 2 mutations ever found in this gene. And while additional functional characterization is still ongoing, these are very promising results. So for this gene, we can say that results from the association study and results from the mutation analysis indicate involvement of this gene in the pathogenesis of obesity. The last gene we studied was a single-minded 1 gene, which is a transcription factor that is expressed in the brain. And chromosomal aberrations leading to a deficiency in SIM1 have been shown to lead to an obesity phenotype in humans. We therefore hypothesized that mutations could also lead to a comparable phenotype, and we performed mutation analysis in our cohort. We found 4 novel coding mutations in our cohort of obese children. However, we also found 4, as indicated in gray here, novel variants in our lean cohort. So in order to differentiate between these variants found in obese cases and lean controls, we performed functional experiments to verify whether or not they affected SIM1 transcriptional activity in vitro. As we can see from these results, for 2 variants which were identified in obese cases, we found a significant decrease in SIM1 transcriptional activity, while for all variants found in lean controls, transcriptional activity resembled wild type. So we can conclude that the threonine 481 lysine and the alanine 517 valine mutation led to a decrease in SIM1 transcriptional activity, indicating that SIM1 mutations can play an important role in the pathogenesis of obesity. For a more comprehensive overview of this study, I would like to invite you to my talk on Wednesday, where the details of this project will be discussed. So taken together, we can say that the leptin-melanocortin pathway is extremely important in the pathogenesis of obesity, because deficiency of nearly every player in this pathway can lead to an imbalance in energy homeostasis, thereby causing obesity. This means that all novel players within or downstream of this pathway should be considered as novel candidate genes for obesity. With this PhD project, we provided proof for several downstream genes to be involved in the pathogenesis of obesity, and these downstream genes should then also be considered as potential novel targets for anti-obesity therapies. With this last slide, I would like to thank all collaborators for this project, especially my promoters, Professor Dr. Wim van Hul and Professor, Luc van Gaal, and of course the Belgian funding agencies that made this research possible. Thank you.
Now the floor is open for discussion. Any questions? One question there. I don't know if we have a microphone.
The mutation of which gene forces a SIMI mutation or associated with mutation observed in leptin or leptin receptors?
So you're asking if there are single mutations or whether they are associated with mutations in other genes? No, we have found these mutations only in SIM1 because the patients were previously screened for mutations in leptin and in melanocortin 4 receptor. So they are single mutations.
It works. I have one question for you. There are also epigenetic changes in these genes?
Epigenetic changes in
mutations in the genes of melanocortin 3 receptors and asfatin 1, and there are also there is a possibility of epigenetic changes despite not having mutation?
Of course, yeah. Well, there's two sides of that story. First of all, there could be epigenetic changes in other obese cases from our cohort that we do not know of. On the other hand, I showed you that there were some SIM1 mutations that had an effect on transcriptional activity and others that do not. So for those that do not, there could be some epigenetic changes, making it gene-gene or gene-environment interactions that could still cause the obesity phenotype seen in the patient. So it's still possible, but we haven't had time to research that.
Any question? So congratulations for this excellent work. And thank you, Dr. Siegers.
Thank you.
Dear Professor Frubeck, dear guests, it is my pleasure to present the second candidate for the Young Investigator United Best Thesis Award, Dr. Claudia Zigorski from Germany. Dr. Zigorski is a psychologist that is currently holding a position as a research fellow at Institute of Social Medicine, Occupational Health and Public Health at the Medical Faculty of University of Leipzig. Working for the largest research center on obesity in Germany, the IFB adiposity diseases, has given her the opportunity to collaborate and work with highly renewed scientists in the field, such as her supervisor, Professor Dr. Steffi Riedel-Heller. She will now show us her presentation entitled The Stigma of Obesity and Its Implications in Public Health. Dr. Zigorski, the floor is yours, please. Could you come? Is it here? Yeah. Okay. Yes.
I think I got it. All right. Thank you for the kind introduction, and thank you for the nomination. I feel very honored today to be able to present the results of my thesis here today. And before I begin, let me introduce the main There we go. Let me introduce the main definitions and terms that I will be using throughout this talk. The term stigma refers to a physical, psychological, or social mark that makes a person different from other members of its group and excludes that person from group inclusion. And stigmatization then is the following process that associates negative adjectives or attributes with that person due to that stigma. And this discrimination then refers to the systematic exclusion and limitation and disadvantages on a personal and societal level. Now, starting with my thesis, I really asked myself, why bother? Why go and research the stigma of obesity? And I think there's really 2 points in really looking into stigma of obesity. First of all, as we all know, we are really lacking effective treatments in obesity. In conservative treatments, we see a weight loss of 10 to 15% in obese individuals, still leaving them obese. So we really are at a point where we cannot make obese individuals back to normal weight. And weight stigma really has a vast impact on the individual's health, even leading to an increased morbidity and mortality, as Poole and Hoyer put this model together in 2010. And it used to be that people would think that discriminating or stigmatizing individuals would lead to a motivation for them to fit back into the society, so making obese individuals want to lose weight. But really what we see is the quite opposite. We can see that in stigmatized individuals, psychological disorders increase, as well as unhealthy eating and eating behaviors and activity levels. So I think this is really an important issue, and let me give you a brief introduction to the model I used during my thesis that was put together by Lincoln Fallon in 2001, and really goes by the terms I just introduced to you. That stigma right here, the difference that we see in obese people, is associated with negative attributes and then followed by subsequent discrimination on an individual structural level, but as well as self-stigma. And really that discrimination forms a new stigma. And for example, discrimination might form another stigma in socioeconomic context, and then really putting that model into a vicious cycle. And the main question in obesity stigma is, why does that mark, why does the obesity is being linked to that negative attribute? And one theory that tries to give an example or tries to give an explanation for that mechanism is attribution theory. Causal attribution theory really puts the role of perceived responsibility into focus, and it basically claims that the higher the perceived responsibility is, that the higher the stigmatization level is. And for obesity, it has been shown in experimental research that it is seen as a self-inflicted, self-controllable condition. And at the beginning of my thesis, we put together a focus group study, which is focus groups are really small discussion groups with participants, and we had participants, obese participants, but also normal weight participants. And it's quite a lot of work to do these focus groups, because you audio tape the discussion, and then you transcribe all the words that were said, and then you analyze the data that you have. But it's quite worth it, because you really get a new perspective on things when you do it properly. And we were really surprised to see some of the results that we found. And I brought to you 2 quotes from those focus groups today. And there's one that a nurse said during the group, and she says, but if one does not think about anything and eats and eats and eats and thinks, why I'm so fat, and then continues eating the next day, well, then it is a vicious circle, indicating that even healthcare professionals have a view of obesity as a self-inflicted condition. Maybe even more surprisingly, obese patient told us basically the same thing. That one patient said, I think it's more an issue of willpower. I claim that every person that's overweight has the initial will to lose weight, but hates himself or herself for not being persistent in the long run. So even obese individuals seem to think that obesity is a self-inflicted and self-caused condition. And as we just heard from the talk before me, this is really just not the case. In the next step, we conducted a systematic literature review to see what representative studies are out there, because I think obesity has become such a societal problem. I think we should focus on representative studies in the general public. And we really only found 7 studies total worldwide. And looking at attribution theory, we were able to find that the lack of activity behavior was agreed on as a major cause of obesity by the vast majority of the general publics that were asked. So we have 82% and then 75% over here in those 2 studies, while genetics were only agreed on by about 1/3, and that was pretty consistent, and it's pretty consistent throughout the non-representative studies as well. So genetics are really not perceived as a major cause of obesity in the general public. So we can conclude that attribution theory, at least the first part of it, saying that obesity is seen as a self-inflicted condition, seems to be true in general populations. On the next step, we wanted to look, well, if is there proof or is there indications that negative attributes are ascribed? And there was only one representative study worldwide that was conducted in Germany, actually. And the authors here found that about 1/4 of the German population showed negative attitudes towards obese people. And they found 3 associated variables, and one of them was causal attribution to the individual. So going in line with theory. Going from that review, we found quite a few gaps in research, actually. For one, we thought, well, so far in those studies, nobody ever differentiated between different kinds of obese people on different ages or different sexes. So basically what we did was we conducted a telephone survey where we called people, and in total we had over 3,000 respondents in that study. And we read vignettes to them, and vignettes are really just fictional descriptions of persons. So we would read out, well, imagine a child, he or she, so we included both sexes. He is 9 years old, and he is 1.46 meters tall, and he weighs 60 kilograms, making him very obese or very, very overweight. And then ask the following questions. And we did that through 3 age groups. We had adults and senior citizens. And then we read potential causes on the as to why this vignette is obese, and asked the respondents to rate these causes. And for stigma, we used the semantic differential, the fat phobia scale, and asked the respondents to ascribe adjectives to those vignettes we just read to them. In order to control for answering behavior, we also included a set of normal weight vignettes at the end of the questionnaire. And then one of the first studies we looked in was, well, what about those negative attributes? And we were able to show that, and you can see that in blue right here, that the overweight vignette was rated more negatively consistently over all 3 age groups, while the normal weight vignette was rated moderately, just, you know, within, as a neutral state. And what you could see from that, what you could see from that graph right here is that the child vignette actually is rated the most negatively compared to adults and senior citizens. And we saw the same thing in a multivariate analysis as well. We saw that overweight people would rate the overweight vignette a little more positive, less negative. We saw that the child actually was, even in a multivariate model, the child was rated far more negative than the adult and the senior citizen. And we were able to show that internal causes, self-inflicted causes of obesity, if you agreed on them more, then you would rate the overweight vignette far more negatively than if you thought that genetics played a major role in obesity. On another publication, we looked into the discrimination level of the model, and looked into what does stigma or what do those negative attributes do on a prevention level. And we were able to show that 98% of the German population actually thinks that obesity prevention is possible, which I think is a good sign to the implementation of prevention efforts in the general public. The majority, almost 60%, would actually partake in those obesity prevention programs. So we can really see an effort to stay normal weight, to prevent further weight gain. And almost all of these that agreed on that they would partake in one of these programs, they would at least partially pay for that themselves. So there seems to be a great willingness to pay in the German general public. So to conclude, public health campaigns really need to encompass a knowledge transfer on the etiology of obesity, and they really need to focus on portraying obese individuals in a positive or in a more positive way. And I brought you one example of the Canadian Obesity Network right here. They actually did a photo shoot and tried to portray obese individuals positively in competent roles and neatly dressed and nice pictures. Because once you look in the media right now, you can find so many stigmatizing images of obese people that I think this is really one major set point for intervention right there. And also, obese children need to be addressed in anti-stigma campaigns. That has really not been pointed out before, but we know that obese children are being stigmatized in schools by their peers, but we found in our studies that they are also stigmatized by other adults. So really, we would have to focus on children on that level as well. And also, the public is willing to finance, at least partially, the necessary prevention efforts, and that really should be taking into account when planning future public health campaigns for healthcare systems. Because I think thinking about making the public or implementing good prevention efforts into the general public, and considering that they might be able or might be willing to pay partially, will give us a chance to actually implement those really costly programs. Those are the 3 publications that went into my thesis. We also did some other research just in attitudes towards obesity surgery, for example, and we were able to find there that the public is still pretty hesitant about obesity surgery. Another point where there needs to be more information, there needs to be more campaigning to transfer knowledge into the general public about obesity. And with that, I'd like to thank you for your attention, and I'm happy to take questions.
Thank you for the excellent presentation. So the floor is open for questions.
Thank you for your presentation. So I was wondering, you briefly touched upon the self-stigmatization. And how prevalent is it? And I think you briefly mentioned something about it, but how prevalent is it within the obese population? That's actually
we really don't know. That's one of the future research projects I'm applying for right now, actually, because there's no really valid numbers on self-stigma yet. We know that it is there, and we have some evidence from other stigma, like racial stigma, that it has to be there, but we don't know number-wise yet.
Right, and maybe a short follow-up question.
Sure.
So you also mentioned that self-stigma can also underlie several psychological disorders. So can there also be like a vicious circle? Because of course psychological disorders also underlie the development of obesity. So can you comment on that?
That's what I think. That's what this research really brought me into thinking. Because once you have a perceived stigma, you try to, or you might tend to self-stigmatize yourself, or you might tend to self-stigmatize, and then those psychological disorders might evolve, and then like you said, making it a vicious circle in the development. And even talking about weight loss programs and the results they really they show, it might be self-stigma that is holding them from being successful. It might be one that.